On me, my name is Sarah Duhlman. I am with Promega Corporation. I'm part of our Pharma Biotech Strategic Business Unit. And it is my honor today to moderate the panel, Looking Past the Pandemic. So to my right, I have Daniel Lawton. He is the CEO and chairman of Promentis Pharmaceuticals. And then I have Professor Dave O'Connor. He's a UW Medical Foundation professor of pathology and laboratory medicine at University of Wisconsin-Madison. And then we have Samir on the end there, one of my colleagues from Promega. He's our director of applied markets at Promega. And so I'll ask each of our panelists to step up to the podium, introduce yourself and your work. And Samir, let's start with you. Ooh, Samir, good intro music.
We're at a disadvantage here, Dave.
Yeah, I know. That's how I roll. Well, thank you for that introduction, Sarah. It's an honor to be here. Grateful for the opportunity for, through BioForward to be able to be a part of this panel with the distinguished panelists here. And looking forward to the discussion today and getting to know some of you here as well. I am thinking that these are our distinguished panelists, including myself. And just a quick introduction about myself. I lead an applied markets business unit at Promega, and we are primarily charged with developing tools and technologies that are being used in the applied markets, such as agricultural food testing, veterinary sciences area and research, as well as environmental testing. And so what I'm going to talk to you about today falls under the environmental testing area, and that is the area of wastewater-based epidemiology. So what is wastewater-based epidemiology, and how does it relate to COVID? Well, this is an approach using analysis of sewage water to identify and monitor biological and chemical materials in the wastewater. And some of you may have heard a lot about this over the last year, and that has really come on the spotlight as the pandemic kicked off and got underway. But it is by no means a new area. It's been used for many years prior to last year, specifically in detecting pharmaceutical waste, the use of drug abuse, like opioids, for instance, specifically here in the US, as well as some countries have used this testing of wastewater very successfully to test for polio and to contain the outbreaks of polio and the likes of hepatitis A. So with the emergence of the coronavirus last year, one of the water research institutes in the Netherlands identified the presence of SARS-CoV-2 in their wastewater in and around Amsterdam, and this was late March 2020. And around the same time, other research groups confirmed the shedding of virus particles in the stool of some of the infected individuals, including those who are asymptomatic. And so if you're looking at that image here in the bottom, so essentially the virus is secreted by the infected individuals, goes through our sewer systems, ends up in our wastewater treatment plants. And before it is starting to be treated with, getting rid of the chemicals and the pathogens, et cetera, it's typically scooped out prior to that and then, tested for the presence of the virus. And then, researchers can calculate, the amount of viral load in genome copies per liter, for instance. And then you can compare that data from the waters to other public health data, such as clinical data, hospitalizations, et cetera. And together, as a data set, try to determine, public health decisions, whether it's on containment or increased testing in a particular region or community, or even, increasing the incentives for vaccines, as it is the case today. So what we've learned is that, the virus is shed in the stool of infected individuals 3 days to a week ahead of time, ahead of emergence of symptoms, rather. And so because of this, the approach of wastewater surveillance has a strong potential to be a leading indicator of new outbreaks. And the image here, the data set on the right, the orange line indicates the viral load in the sewage water as it was tested by that group in Netherlands from sometime in February to May. And they showed that the spike in viral load in sewage water happened about 7 to 10 days ahead of some of the hospitalizations that it can be shown in the gray line. So, again, this makes it a very powerful approach in managing the pandemic, or one of the tools in managing the pandemic as we move forward. So currently, there are about 57 countries globally testing for coronavirus in their sewage. So it's a global effort. Most of the countries in the map here that are in some shade of orange or red are testing for coronavirus. And here in the United States, 37 states are working with the NWSS, the National Wastewater Surveillance System, implemented by the CDC. And many others are also testing through private services, et cetera. The state of Wisconsin is also testing their wastewaters in multiple different municipalities, 70 plus, I think. And that is undertaken in collaboration with the Department of Health Services, Wisconsin State Board of Hygiene, and University of Wisconsin. So this testing is primarily undertaken by public health, academic, and commercial service labs. And as I mentioned, this is something that has been already happening, but has come really to light and the global stage because of the pandemic. And what's really exciting is that we've shown that this approach can really help us determine where to focus clinical efforts, whether things are trending upwards or downwards. And moving forward, it's nice to see the commitment from some of the federal agencies like the CDC in the EU as well as NIH, really putting funds to put together a sustainable national surveillance system. So I'm really excited to introduce that, and looking forward to the pandemic. The CDCs and the likes are thinking about using this approach for other pathogens as well as we look forward in the pandemic. So with that introduction, I'll stop here and ask David to come over.
Hi, everyone. I'd like to thank the organizers and thank the excellent presentation we just saw. My name is Dave O'Connor. I'm a professor at UW-Madison. Show of hands, how many people are sick of the pandemic? Okay, most of you. This is okay. This is normal. And I say this because I've spent most of my career as an HIV researcher. And back in 2012, I remember that I was in rural Uganda, and I was talking to someone on the street about HIV, just random conversation, a guy on a motorcycle, and saying, gosh, how do you deal with it? HIV was really common in the area where I was. How do you deal with it? He goes, I just don't think about it. I couldn't really wrap my head around it at the time until a couple years later. I was down in South Africa in a taxi. I asked the same question, someone else, and they pretty much gave the same response. They were just sick of it. And so I think when we think about what's going to happen after the pandemic, we need to bake pandemic fatigue into whatever our thinking is. And that pandemic fatigue, we're seeing it now. We see it every day in the demonization of people who work in public health. We see it in the way that masks have been politicized. As a prelude, we are absolutely going to have the same, it's like you can just rewind the VCR tape, and we're going to have the same questions about third doses of vaccines and what constitutes someone being fully immunized. If you have the opportunity, by the way, to get a third dose of a vaccine, one of the mRNA vaccines, or when the J&J second dose becomes available, I'd strongly encourage you to do so, because what we know now is that those vaccines really probably should be delivered as a 3-dose series for the mRNAs, with the third dose being several months later, because that's how we increase our protection against the Delta variant. It's how we deal with the fact that the pandemic is changing in front of our eyes, and the circumstances as they were a couple of months ago are not as they are now and are not as they will be in a couple of months. And so we're going to be sick of the pandemic, but just because we're done with it doesn't mean it's done with us. And so when we think about what's going to come in the future, we have to be thinking about lightweight ways of doing surveillance, so lightweight ways of doing monitoring, ways that we can integrate protection and prevention into people's daily lives. Wastewater sampling is one way of that. As the kids' book says, everybody poops, and as long as that's the case, we're going to be able to do surveillance without people even knowing that we're doing it. One area that we've been moving into, I've been very involved in school testing around Dane County since the last academic year. There's a flood of resources that are available now that were not available for testing in schools this time last year. In a sense, that's really, really great. In another sense, though, it does trade one set of problems for another, because you now have a massive amount of testing going on, but that leads to a massive amount of reporting and a massive amount of contact tracing. And as we get into the winter, it's going to become more and more difficult. Right now, the problem is kids with seasonal allergies are conflated with kids who have COVID-like symptoms. As we get into the winter, it's going to be kids with RSV, influenza, other upper respiratory illnesses that are going to be conflated with COVID. And you're going to have to surge even more testing and deal with even more kids being positive for other viruses, but getting swept up into sort of a COVID cyclone if we continue the course. And so there are innovative ways that we can be thinking about that, whether it's using quick antigen tests in preference to the more sensitive but more difficult to implement PCR tests, or if it's other types of environmental surveillance. So we've been working a lot on air sampling in schools. So here in Dane County, we have a network of K through 12 schools and other sites that are running aerosol scent samplers, which are made by Thermo Fisher. I don't know if we have any Thermo reps in the audience. Thank you. It's an awesome tool. And it basically sucks in the air, so that if I was running one in here, at the end of the day or at the end of this session or whatever interval you want, we could pull a cartridge out of it. We could then sample that cartridge for SARS-CoV-2 genetic material, as well as genetic material for other viruses. And we could score the room, and we could say, okay, on the balance, was this room at low risk, moderate risk, high risk for having a lot of SARS-CoV-2 genetic material? Everyone's wearing a mask. That's great. That's also not going to survive forever. We're just not going to be able to sustain masks and the heightened risk mitigation that we have now forever. And so when we think about what is going to come post-pandemic, I think we need to plan for an intermediate place, which is pandemic fatigue pandemic, where we still have a lot of work to do, where there's still a very high level of risk. So Dane County, where we're standing right now, is in a high community transmission risk zone per the CDC. We had 400 cases on the Public Health Madison-Dane County dashboard yesterday. We've been inured to seeing those sorts of statistics every day, but that doesn't make it normal and it doesn't make it safe. And so that's what we have to be kind of thinking about, is how do we return to some sense of normalcy, understanding that people are going to be exhausted. They're not going to want to think about the pandemic, but that doesn't mean that the risk will have gone away. And so these are the kinds of things that we're thinking about. And so with that, I will turn it over to Daniel. Thank you.
You know, everybody has been doing such a nice job with masks. I'm going to go ahead and speak through this guy and keep my mask on. Is that cool? Hi, everybody. Thanks, David. Thanks, Samir. Really fascinating stuff. It only really reinforces to me what an extraordinary challenge it's going to be to fit all of the material we have within the short period. Sarah, you have a challenge in front of you. I am the CEO of Parentis Pharmaceuticals. We are targeting a novel and very exciting target in the brain called XC-. We've developed a portfolio of molecules that very selectively activate that target. We have gotten our lead compound, which is 2023, through 2 phase 2 trials, the first of which we refer to as a RECON study, which demonstrated that 2023 gets into the brain and 2023 activates the circuitry that we're targeting, which is focused on impaired executive control and impulsivity. The second study is the first ever multi-site study focused on trichotillomania. a disease that, although I suspect some of you have some experience with, most people find difficult to pronounce and everyone finds very difficult to spell. But we sometimes refer to it as the most common disease no one's ever heard of. It is compulsive involuntary hair pulling. It is far more serious than we would have ever imagined. An important first indication for us. We've shown that in an important majority subpopulation of trichotillomania patients, our drug enables executive control over the behavior and reduces the pathological behavior. With these results in hand, we're very excited about our second phase 2 trial in trichotillomania, but we're also equally excited about a whole range of other indications. And if you think about what I just said, it probably makes sense. If we can enable executive control and diminish the power of impulsive behavior, which we can do, and we've shown we can do in a number of studies, there are a great number of indications that suddenly become very interesting. So binge eating, alcoholism, drug abuse, maybe cognitive impairment associated with Parkinson's disease, et cetera, et cetera, et cetera, maybe PTSD. And maybe, so among all that mix of potential follow-on indications, go to COVID and think about not acute COVID, but long COVID. Long COVID, which is really a very difficult to define, and we'll maybe talk about this if we have a chance, just how difficult to define it is and how amorphous it is and how heterogeneous the patient population is. But one of the hallmark features of long COVID is cognitive impairment. And so when we look at where we should be focused and where it makes sense to apply these drugs that we've developed, long COVID is obviously one of the targets. Not only because of the cognitive element that I just described, which is probably obvious, but also because of the fact that our drug acts both within the CNS, within the central nervous system, and peripherally. That's got to be a necessary precondition of a successful therapeutic for long COVID. Also, we act, and we'll not get into this detail mercifully, but we act against inflammatory mechanisms and act against oxidative stress mechanisms, both of which are going to be very interesting and very useful in the context of COVID. I think that's probably it by way of intro. If we're not careful, we're going to lose our time for questions and answers. So delighted to be here, delighted to be part of the panel, and Sarah, I'll hand it over to you.
All right, thank you. Thank you, everybody, for those introductions. We would really love this panel discussion to be a dialogue with everyone. So if you have questions, please let us know. In this room, because it's smaller, we are just raising hands and asking you to speak up. So does anybody have a burning question that they would like to start with? Okay, luckily. There we go. Go ahead.
So this question is for Samir. How tightly dialed in can that wastewater sampling be? Like, can it be to a building level for, say, monitoring individual dormitories? Or, instead of waiting until it's immediately at the waste treatment plant to assess the community?
Yeah, thanks for that question. What's your name?
Sarah.
Sarah, excellent question. We've seen a variety of different strategies from the municipality level. There's been primarily taken from the wastewater treatment plants as the water makes their way there. And that sort of helps the community, get a snapshot of the community in that area to see if the viral levels are going up or down. Certainly upstream testing is done and has been shown to be effective. So campuses, as you mentioned, this is one of the areas that the wastewater-based testing can be really, really powerful because there has been numerous examples right here in the US, University of Arizona, Utah State, and even here in Wisconsin. They did quite a bit of clinical testing as entering, certain population came into the fall last year, and then continued the wastewater testing within each of those dorms. And sometimes, depending on where you are on the infection kind of timeline, you can definitely miss it. And what they did was they picked up an increase in RNA levels in the wastewater from a couple of dorms, And lo and behold, they retested the population and found that there were a couple of asymptomatic students that had come in. So really, kind of a 1-2 punch to really understand what's happening at that level. And certainly we've heard of hotels, we've heard of senior homes, et cetera, hospitals also testing for their wastewater. And I think it can be scalable across the different parts of the spectrum. Thank you.
Thank you, Samir. Yeah, question.
I'd be interested in hearing anybody's perspective as to how long we'll be dealing with COVID, both the current pandemic and longer term. Is there an expectation that people will be encouraged to have an annual vaccine like along the lines of influenza?
Dave,
All right,
you're-
people are looking at me. Okay, so so the answer to the first part of the question is that we need to be gearing up that this is going to be a multi-decade fight. As much as people hate to say that, HIV was, the first AIDS cases were found 40 years ago this year, and we're still dealing with it. It's still a major global problem. In terms of a day-to-day problem, however, the vaccines are going to be a pathway out. The exact cadence for boosters is yet unclear. We need more data. We need basically to figure out how the immunity decays over time. Some of the data that's out of Israel, as well as some of the early clinical trial data from Moderna and Pfizer, strongly suggests that after the second dose, so what if you got an mRNA vaccine earlier this year, you might have an immune response that's like 10 arbitrary units. After you get a third dose, after 5 or more months, you'll have an immune response that's 50 or 100 arbitrary units. So that third dose increases the amount of neutralizing antibodies much, much higher than after the second dose. What does that mean? Well, imagine you're a helicopter flying over an ocean. The first couple of doses keep the helicopter above the surface, but if there's a big wave, it could still get submerged. Now, with these third doses, you're a couple hundred feet clear, and so as long as you're starting at a higher point, it's going to take longer for those immune responses to wane. And so it might be that you don't need a fourth dose 6 months from now. Maybe you would need it 3 years from now or 4 years from now. But again, some of this depends on what happens with the virus itself. The assumptions that we made about vaccine duration at the end of 2020 and the beginning of 2021 didn't factor in Delta. Didn't factor in a much more contagious virus that had some vaccine escape. What are we not factoring into our considerations today about how the virus is going to look this time next year? So I would say I think there is promise that a 3-dose vaccine will become the standard, kind of like hepatitis B vaccine, but with the big asterisk that the virus still may have curveballs to throw at us. Sure, so the question is, what about antivirals and what about treatments? I think that's going to be an important piece. I think that the big challenge with treatment is that you have to get people early enough in the infection for the antivirals to have a strong effect. I think the new Merck drug looks very promising. As someone who comes from an HIV background, I cringe just a little bit when you're talking about monotherapy because that could breed and engender resistance in the virus, given enough time. We'll just need to see. I think it is absolutely true that an ounce of prevention is worth a pound of treatment. And if we can prevent people from becoming infected at all, if we can prevent them from developing severe illness, that's way better than trying to treat people because that's always just going to be expensive and hard.
What is the best way to test for COVID?
Anyone else? I can keep going, but I want to make sure I give...
No, your best position to answer.
Okay. So best is a loaded adjective. The most sensitive way to test for COVID is with the PCR tests that are quite standard. In an idealized world, if there was unlimited resources and immediate turnaround, that would be the standard that we would want to use all the time. Unfortunately, we're not in an idealized world. So the antigen tests that you can get at Walgreens or CVS are a little bit less sensitive, but they're a lot more convenient. They're much less expensive, and they're much more immediate. And so I think that in most settings, the antigen testing in this country has been vastly underutilized because of this perception that it's not as good as PCR. But the reason that that perception has taken root is because people are comparing idealized PCR testing programs to idealized antigen testing programs, but that doesn't translate to the real world. So for example, in schools, I think you would get a lot more mileage out of testing kids with antigen tests on site, continuing risk mitigation with masks, and if kids are close contacts, but if they test negative on an antigen test, assume a little bit of risk that the antigen test might be missing someone, test them again the next day, but let them stay in school. Don't send them home for 10 days while you try to figure it out with a molecular test and err too much on the side of caution, because there's a downside risk there that you're being so disruptive to schooling and education that kids who have seasonal allergies and a couple of colds are going to miss 30 or 40 days of school this year, and that's not a great outcome.
Dave, I'm inclined to generalize a little bit, or I guess I'm inclined to ask you whether we can generalize a little bit from what you've just said, that in this years-long, or indeed decades-long struggle that you've described, that perfect might well be the enemy of the good, and that we might well have to satisfy ourselves with good but not great solutions across the board. Is that a fair generalization?
Absolutely.
So, yep.
Sort of a follow-on to the first question from Sarah. As we think about deploying surveillance and thinking about it as almost like infrastructure, stop and go lights, how do we push that as a community to happen? Like, who needs to buy in and what are the barriers to get it there?
What's your name?
Justin.
Justin. Yeah, that's the question du jour. In terms of, we know that this approach works. It's extremely valid. The question now is, how do we make it sustainable so that we can catch the pathogens, whether it's SARS-CoV-2 or something else down the line, earlier so we can do something about that? I think the ingredients of that are coming in place. Of course, the funding is coming, resource development, capacity building. That is slowly emerging through the public health systems, through the academic centers, and the departments of health. I think one of the barriers that we need to think through is the communication between all of these stakeholders, and really to have a much more regional or community-based action plan. So if if the virus levels are going up in Appleton, what does that mean? Like, how does that, work together with the Department of Health Services, and what can we do about that? I think those questions still need to be figured out, and who ultimately is going to be the driver of action as a result of what we're seeing from the data standpoint. So certainly things that are still need to be worked out.
Dave, do you have any comments from the air sampling aspect of that?
It's an exact mirror. It's the exact same thing. It's how do you couple the data with action? And you can imagine, if you had surveillance in a hotel, to build on Samir's example from earlier, is the hotel going to be willing to close? Are they going to want more visible precautions? One of the real barriers that we've seen, we've been putting these air samplers in different places around the region, and one of my colleagues in Minneapolis just emailed me this morning and talked about how a number of the schools and a number of the places that he approached to put them don't want the air samplers because they don't want to know. Because they don't know what they would do with it, and if they get bad news, sometimes not knowing is better than knowing and having to deal with something you don't want to deal with. And that's, I think, going to be a big problem for all these sort of sample-independent surveillance technologies. How do you convince people that it's worth their while to know if there's SARS-CoV-2 in the wastewater or in the air, if that's just going to create inconvenience or cause them to lose business or make their lives more difficult in some other way?
No, we had another question over there. Mm-hmm.
Yeah, I'm wondering if someone can provide a little bit of insight into ivermectin and the effectiveness versus ineffectiveness for treating COVID.
I keep looking at you, Dave.
Okay, I can do it. As those who have followed that story know, ivermectin was first shown to have some in vitro activity against SARS-CoV-2 in Australia early last year. As a fun bit of trivia, I actually lived in Australia in 2018, 2019, and I'm good friends with the person who was the lead author on that study. He was really excited about it when the data first came out. And when there was follow-up data from an Egyptian study, it was small, but it was provocative. Unfortunately, some of that data looks like it was falsified. And so what you have is you have a drug that is used in people. If you're using it appropriately in people, I'm saying not using like the veterinary product, but using the product in doses that have otherwise been used in people, I don't think it's going to cause that much harm, except the opportunity cost that you're not using drugs that are actually going to work, whether they're monoclonal antibodies or remdesivir, or if you're really sick, high-dose steroids. And I think it's a sign that it's gotten so politicized that people want ivermectin to the exclusion of all other things, or people just look at it as, a horse dewormer, when the truth is that there is no great data that says that it works. And so if it were me or my family member, I wouldn't want them to get it. But I think that, there's likely going to be drugs that work against SARS-CoV-2 that are going to come from unexpected places. And so we shouldn't be dismissive of the idea that drugs might come or from repurposing activities. I think in this particular case, even my friend Leon, who did a lot of that seminal work, would acknowledge that as excited as he was when he first did it, the data that's come to light since then has not substantiated it, and has not substantiated the sort of excitement that it's galvanized here in the US in the last couple of months.
So let's take a second to talk a bit more about therapeutics. So we're talking about acute COVID here. Something, Daniel, that you brought up is these long-term COVID effects. And, what are we seeing there? What do we know? And then when we go into the drug discovery realm and clinical trials in that space, how is that going to play out?
Well, what we've seen is an extraordinary collection of symptoms which center around a number of common elements, not uniformly common elements, but common elements, one of which, as I mentioned before, is cognitive impairment, what's sometimes called brain fog. I suspect, so maybe stepping back a second, what you need when you're developing a drug is a known condition with known measures by which you're going to assess that condition, an understood population who will be assayed, including who will be excluded from the population in question and who will be included in the population in question. And the challenge with long COVID, and the challenge that we're dealing with in considering the application of our drug to long COVID, and particularly cognitive impairment in long COVID, is we really don't know any of those things. The work that I'm most impressed with regard to long COVID suggests that it's probably more than one thing. It certainly includes a vast difference in population. It includes people whose acute COVID symptoms were forgettable, were trivial, and who go on to develop these long COVID symptoms. It also includes people who had massively dramatic acute symptoms, people who were intubated, people who were in ICUs, people who experienced organ failures. So the real challenge, at least with regard to cognitive impairment as it's implicated in long COVID, and it's a big deal. It is one of the hallmark features of long COVID, but is figuring out those threshold questions without which it's really, really difficult to run a trial and really, really difficult to develop a drug. I don't know, Dave, what you'd add to that.
No, I think it's spot on. I mean, the virus does get into multiple organ systems. The virus can, in some cases, get into the brain. So sometimes it might be a direct virus. It might be, if you're hospitalized, it might be post-ICU, post-vent brain fog, and it can affect lots of different systems. And unfortunately, medicine is not the greatest at dealing with diffuse, multi-parameter types of conditions like this. And so I think it's also very, very common in unvaccinated people. It's estimated that up to a third of people continue to have some manifestations of infection at least 12 weeks later. We're kind of some people, a friend of mine has not had her sense of smell back for more than a year. Is it going to come back at some point? Who knows? No one's had it for 5 years to know what the 5-year, 10-year, 20-year complications are. But it's going to be really, really tricky for all the reasons Daniel said.
I'm wondering your thoughts on moving forward. The pandemic really highlighted health equities in prevention and treatment. What would you like to see moving forward in your prevention strategies and or treatments to address health inequities?
Yeah, I mean, I think it's really brought home the health inequities here in the U.S. The hospitalization rates are not equally spread. Some people have been essential workers in harm's way throughout the entire pandemic. Others are lucky enough to spend a lot of their time working from home and being largely protected by circumstance. I'd like to think that actually long COVID might, in a paradoxical way, kind of help bring to the surface this idea that it could happen to anyone, that those care costs are going to be expensive, and that medical coverage and access to health care needs to be available to everyone, not just for long COVID, but with long COVID as sort of being the leading edge of that spear. Really recognizing that when there are these health inequities, not only does it put a disproportionate burden of disease on certain subgroups that are heavily affected, but it affects the entire communities. So even if you don't particularly give a whit about the health inequity for its own sake, you should selfishly care about it because risks in another community affect your own personal risk when you live in a community. This occurs within a small municipality, it exists within a state, a country, and it also exists around the world. The Delta variant that emerged and is the only variant we see in the US was first detected in India. They had an apocalyptic, catastrophic surge back in May. Had that surge been prevented, had the next surge of that sort be prevented through vaccination and high access to vaccines in Lagos or wherever the next one is going to come from, that provides protection to us here if we can prevent huge burdens of disease happening elsewhere. So I hope it shines a bright light on it, but it's going to be tricky.
Yeah, in addition to David's comments, I think that this is particularly the inequities area is where the surveillance can come into place, where it's a pretty cost-effective way to monitor a particular community or region. So I was happy to see that there's a line item within the recent American Rescue Plan where they're talking about funding the surveillance, which includes potentially wastewater testing, clinical surveillance, other types of surveillance, animals, etc. And you can see from this map here in the globally, right, like where the infrastructure is starting to come into place, and you can easily pinpoint the white areas. So, however, having said that, I think the wastewater surveillance is definitely an area that can mitigate the risks of the inequities. And I know that the National Wastewater Surveillance System by the CDC has really emphasized on areas around the indigenous population where folks don't have access to healthcare or may not necessarily think that they need to be able to get to a hospital or a clinic for testing. So that's just something that came to light.
Samir, from the funding that you've seen, what are some of the responses to, let's say we do the wastewater treatment in certain areas, you find a huge spike somewhere, what is the response going to be when that happens, or what are your thoughts on that?
From a public health standpoint?
Yeah.
Yeah, I mean, I think, that's where things are a little bit more regional. Different states have different, strategies in place. I know some states are a lot more evolved in how they communicate with the Department of Health, local Department of Health, and how that kind of translates into more targeted testing or more emphasis on vaccinations, et cetera. So I think that it's going to come from a consortia of local health authorities, public health labs, et cetera. I think the federal agencies can provide the canvas in terms of method recommendations, in terms of being able to provide funding. But I think a lot of that is going to be very regional and local.
So we're coming close to time here. Maybe a couple more questions. I see one here in the middle, if you want to go.
Is there any thought to, like with virus mutation and also our immunity, where the COVID transmission from a viral standpoint, and once it transmits before it kills the person, that the symptoms would come down to probably like a baseline where we sort of tolerate the cold or the flu?
Yeah, so I think the question is, are we going to basically co-adapt with the virus so that eventually the virus is mild? I think there is a possibility that something like that could happen. What we have seen, though, is that our expectations about how this virus is going to evolve have been wrong consistently throughout the epidemic. We initially thought that because the coronaviruses have a proofreading activity when they replicate themselves, they have an unusually large genome for an RNA virus. They're about 30,000 base pairs long. That has been enabled by the fact that its replication machinery can error correct, basically. That led a lot of people to think early on that you wouldn't see very much by way of viral mutation, that we would have fingerprints that we could use to track outbreaks in the sequence, but not much else. Then we had this big shift at the end of last year when suddenly we were finding individuals who had way more mutations in their virus than you would have expected based on that sort of stepwise progression of mutation accumulation. We now think that that was probably from people who were immunocompromised, who were infected for a long duration of time, and that allowed the virus to adapt specifically to that person's immune system and to that host. And then when that virus got out and started spreading, it sort of was a sea change in how the virus behaved. And we saw the same thing happen with viruses out of Europe, out of South Africa, out of Brazil, and elsewhere. So as long as the virus is continuing to evolve unpredictably, with a lot of that happening in people who are immunocompromised, it's really hard to say what the next viral trajectory is going to be in terms of how it's going to mutate. Is it going to, it's possible that the next version is going to be extraordinarily contagious and it's going to displace Delta, but it's not going to be very severe. That would give you the sort of phenotype that you're talking about. It's also possible that for whatever comes next is going to have to be at least as contagious as Delta if it's going to displace Delta, but for it to be as severe or potentially even worse. It's important to remember that other coronaviruses, such as the MERS coronavirus, have a case fatality rate approaching 30%. So the possibility that you could have a coronavirus that is as contagious as Delta, but quite a bit more severe, still does exist. And I don't think that's a likely outcome, but I do think it's a rare, an unlikely but high consequence event that we have to be mindful of.
And boy, does it underscore the point you made earlier about sort of informed, self-interested, ethical behavior, both domestically and internationally.
All right, let's do one final question here in the front.
With the mutation rate that's going on right now, does it make sense to have people get different vaccines for their booster to maybe present a different target or go after a different target, or does that open up problems maybe with multi-drug resistance?
Sure, so it's a really good question. The vaccines all are targeting the same part of the virus. All three of the vaccines that we're using target the same part of the virus, so it's not really going to make an effect there. What we do know from other virus systems is that sometimes getting multiple different types of vaccines tickling your immune system, what we call a heterologous vaccine regimen, can actually lead to better immune responses, higher quality immune responses. There might well be value in that. The good news is that some of the best data in the world that's coming out of Israel shows that with the Pfizer BioNTech product, a third dose of the same product is a huge immunologic upgrade. In both the Moderna and the J&J studies that have been submitted to the FDA, they have shown that there's a huge immunologic upgrade with these remote boosts. So I don't think we need to necessarily consider different products from the purposes of avoiding resistance. We might do it because it could give even somewhat better immunity, The more important reason to do it is practically, that I think we have to worry about, you know, if everyone needs to get immunized multiple times and you have to keep track of what product you have and what product is on the shelf and what pharmacy and when, and that becomes a big old mess. It becomes a lot easier if you're able to just show up like you do for a flu vaccine and not worry about what brand of flu vaccine you're getting each year if we need something like an annual booster.
All right, I encourage you, if you had questions that didn't get answered, please track down our panelists in a networking session and have a conversation with them. Thank you all for joining us. Next up is lunch back downstairs on the first floor, and please join me in thanking our wonderful panelists.